Hi, we meet again in biology class. I am teacher Yumni, and today my lecture topic is still dark reaction. Okay, so dark reaction involve of a Kevin cycle. Okay, if you can recall, what is the significance of Kevin cycle in the dark reaction? So it is for formation of sugar from the carbon dioxide fixation from the air. Okay, so if you remember this first stage in the carbon cycle, it is a carbon dioxide fixation. Okay, here it is the action of enzyme Rubisco or Rubisco enzyme in carbon fixation. Rubisco catalyzes the carbon fixation of carbon cycle in C3 plants and produces a 3-carbon sugar. If you can see from here, RUBP, okay, was fixed with carbon dioxide. So it is RUBP 5 carbon compounds plus 1 carbon compound. So it's become 6 carbon compound and it splits into 2 G3P. So total of 2 G3P is a 6 carbon compounds. Now, okay. However, Rubisco is not loyal to the carbon dioxide. Yeah, maybe I don't know, not loyal to the carbon dioxide because Rubisco also has a high affinity towards oxygen. Okay, so Rubisco has high affinity for oxygen. Okay, when does this happen? Okay, when during hot, bright days, stomata will close. Okay, stomata will close to prevent the transpiration. To prevent the water loss via transpiration. So carbon dioxide concentration begins to decrease in the air spaces within the leaf, and the concentration of oxygen released from photosynthesis begins to increase. Remember, light reaction produce oxygen. So the declining level of carbon dioxide in the leaf starts the Calvin cycle. When carbon dioxide CO2 concentration declines, Rubisco adds O2 oxygen to RUBP in the Calvin cycle. So this will result in the splitting of one G3P into, and another one is 2-carbon compound phosphoglycolate. So photorespiration will result splitting of 3-phosphoglycerate into a carbon phosphoglycolate, okay, which is a 2-carbon compound and carbon dioxide. All right. Sorry, it's not carbon dioxide. It's 2-carbon phosphoglycolate and 1 G3P. All right, so this results in the loss of RUBP. Okay, so will result the loss of RUBP. Actually, RUBP, which could be used in carbon dioxide fixation to form 2 G3P. However, okay, because of this phenomenon called photorespiration, so due to the photorespiration, photosynthetic output is decreased. G3P output is output for the photosynthesis decreased since no ATP is generated. So do you think photorespiration is beneficial to plants and why? Okay, so because it reduces produce photosynthetic activity. So photorespiration is not beneficial to the plants. All right, so we continue. As we know that Rubisco is not an efficient enzyme for fixing carbon dioxide because it has high affinity to oxygen also. This inefficiency is overcome by the enzyme PEP carboxylase. Okay, so from the diagram here, Okay, now enzyme is PEP carboxylase. Now, PEP carboxylase has no affinity towards oxygen and has great affinity for carbon dioxide. So, PEP carboxylase is present in some plants such as C4 plants and CAM plants. Okay, so now we continue. We just discovered this new enzyme, PEP carboxylase, to overcome the photorespiration. Okay, in order to minimize photorespiration, some plant species adopt different modes of photosynthesis. They are C4 plants and crassulacean acid metabolism CAM plants. These plants differ in their anatomy and physiology. Right now, we will look at anatomy of the C3 plant first. Okay, if you can see here, C3 plants have two types of mesophyll cells, namely palisade mesophyll cells and spongy mesophyll cells. The spongy mesophyll cells are more loosely packed compared than palisade mesophyll cells. So, if you notice the chloroplast abundance in the mesophyll cells, all right, and in the gut cells. So where does sugar synthesis take place in citrullary plants anyway? It is in the mesophyll cells because mesophyll cells contain chloroplasts and Calvin cycle happens in the stroma of the chloroplast. So now we will look at C4 anatomy plants. Okay, C4 plants have distinct Schiffer cells around the vascular bundle of the leaf known as bundle sheath cells. If you can see bundle sheath cells. These bundle sheath cells are packed with chloroplasts. The arrangement of chloroplasts containing cells around the vascular bundles in C4 plants refer as Crohn's anatomy. Okay, I already moved my cursor to observe the Crohn's anatomy. So we call this arrangement nice arrangement as a Crohn's anatomy cells. Okay, but all right. So now we continue. So of course you can see from here chloroplast in mesophyll cells and bundle sheath cells. Okay, C4 plants have two distinct types of photosynthetic cells such as bundle sheath cells and mesophyll cells. Okay, then where does sugar synthesis take place in C4 plants right now? So as I said, it happens in the stroma of chloroplast. So sugar synthesis takes place both in the mesophyll cells and the bundle sheath cells. So where does carbon dioxide fixation takes place in C4 plants? Okay, here now carbon dioxide fixation takes place in two places, first in the mesophyll cells and then in the bundle sheath cells. Okay. C3 and C4 plants show physiological difference in the opening and closing of stomata pores. C3 plants open their stomata during the day, while C4 plants open their stomata during the night to prevent water loss due to transpiration. So the rate of transpiration from the plant affects the opening and closing of the stomatal pores. This directly affects on carbon dioxide fixation and photosynthesis. So in C4 plants, when stomata are closed during hot conditions, enzyme PEP carboxylase fixes carbon dioxide in mesophyll cells, form a 4-carbon compound malate, which is transferred to the bundle sheath cells. In the bundle sheath cells, malate releases carbon dioxide, CO2, which is assimilated by Rubisco and Calvin cycle as usual. This reduces photorespiration, hence enhances sugar production. So let us see how C4 and CM plants fix carbon dioxide. So I show you C4. So you need to observe C4 cycle. Okay. Okay, if you notice, the C4 cycle, which occurs in mesophyll cell and bundle shift cells, is also called H-slag pathway. The cycle takes place in C4 plants where a 4-carbon compound is formed after CO2 fixation. C4 plants. Okay, because we said C4 plants because the first product form is a 4-carbon compound after CO2 fixation. All right. In C4 plants, CO2 fixation occurs in mesophyll cells, and the formation of sugars occurs in the bundle shift cells. Okay, so stage 1 in the H-slag pathway. Okay, this is carbon dioxide fixation in C4 plants. When stomata are closed during hot conditions, enzyme PEP carboxylase fixes carbon dioxide in mesophyll cells. So what happens to carbon dioxide molecule, CO2 molecule? Okay, first CO2 molecule combines with phosphoenopyruvate PEP3 carbon compound in the mesophyll cell. Okay, how many carbon atoms in PEP? If you can see from here, it is 3 carbon compound and plus plus 1 carbon dioxide. So they will produce 4 carbon compound oxaloacetate. 1, 2, 3, 4 now. Okay. When fixed with the carbon dioxide, they will form 4 carbon compound oxaloacetate. How many carbon atoms are present in oxaloacetate? It is a 4 carbon compound in oxaloacetate. What is the enzyme that catalyzes the reaction? So the enzyme involved now is a PEP carboxylase. Does PEP carboxylase fix oxygen? No, because PEP carboxylase has less affinity for oxygen. So stage 2 in the C4 or H-slag pathway is the conversion of oxaloacetate into malate. Oxaloacetate is converted into malate, which enters the bundle shift cells. This malate releases carbon dioxide and then which is refixed in the carbon cycle by Rubisco. So here, conversion of malate into pyruvate after releasing the CO2. What happens to the malate in the bundle shift cells? Okay, from oxaloacetate need to rearrange to become malate because malate can enter the bundle shift cells. So once malate enter the bundle shift cells, the malate is converted into pyruvate by releasing carbon dioxide or CO2. How many carbon atoms present in pyruvate after release of 1 carbon atom? So it is 3 carbon compound of pyruvate. What happens to CO2 in the bundle shift cell? So as usual, CO2 will fix with RUBP to begin the Calvin cycle, resulting in the formation of sugar as we learned from the previous lesson. Okay, so the enzyme in the Calvin cycle is a Rubisco. Okay, but no worries. Seed plants also have Rubisco, which is present in the bundle shift cells. So this fixes CO2 in Calvin cycle as usual. but no oxygen in bundle shift cells. Here, no oxygenation of RUBP occurs because the bundle shift cells are far from the stomata. So how does it affect the photosynthetic output? Of course, photorespiration is reduced and sugar production is enhanced. Okay, now PEP carboxylase present in C4 plants is a more efficient enzyme compared to rubisco that present in C3 plants because it reduces the photorespiration and increases the photosynthetic output. So now, what happened to the pyruvate? Okay, this is a conversion of pyruvate into PEP. Pyruvate into PEP. This pyruvate re-enters the mesophyll cell where it is converted back into PEP with the addition of one phosphate group. Okay, then where does the phosphate group comes from? So pyruvate to become PEP consume phosphate from ATP. So PEP three-carbon compound combines with another molecule of CO2 to begin the next cycle with the help of PEP carboxylase enzyme. Okay, so apart from C3 and C4 plants, other plants follow a different mechanism called crassulacean acid metabolism CAM plants. This cycle was first discovered in plants of the family Crassulaceae. Okay, now I click on CAM to observe CAM carbon fixations. So CAM plant Crassulaceae, example is pineapple, cactus, right? So in CAM plants, CO2 fixation occurs via the formation of malic acid. Okay, the same way with the C4 plants. This mode of CO2 fixation is called crassulacean acid metabolism or CAM. Examples of CAM are here cactus and a pineapple. Okay, CM plants use crassulacean acid metabolism, another photosynthetic adaptation in arid conditions. CM plants open the stomata at night and initially fix CO2 into organic compounds using C4 pathway. These organic compounds accumulate during the night and are decarboxylated through the day to yield high levels of CO2. During the day, the high level of CO2, all right, drive the Kelvin cycle and minimizes photorespiration. CM plants are similar to C4 plants in that they use both C3 and C4 pathways. However, CM plants differ from C4 plants in that they use C4 pathway at night and C3 pathway during the day. Both pathway occurs in the mesophyll cells. Okay, now in CAM plants during the night, CO2 is converted into organic acid, all right, which releases CO2 for the Kelvin cycle during the day. Okay, so now that's all. The pathways for the C3 plants, C4 plants, and CAM plants. So now we should compare them, right? So if you can see C3 plants, CO2 acceptor is a RUBP, while in C4 plants is a PEP. While CO2 acceptor in CAM plants also PEP because CAM plants use C4 pathway but in at a different time. So enzyme for Rubisco Enzyme for enzyme Rubisco in C3 plants. In C4 plants, enzyme PEP carboxylase, and in CAM plants also PEP carboxylase. Then because they use CAM plants use a C4 pathway to fix the carbon dioxide. And first product in C3 plants is phosphoglycerate, while C4 plants oxaloacetate for carbon compounds, and CM plants also same with the C4 plants. Location of the in the C3 plants, CO2 fixation occurs in mesophyll cells, while in C4 plants, CO2 fixation occurs in mesophyll and bundle shift cells. CM plants, okay, CO2 fixation occurs in mesophyll cells at different times. Okay, so now photorespiration in C3 plants lowers, yes, photorespiration occurs in C3 plants that will lowers photosynthetic efficiency. Now in C4 plant lack of photorespiration, CAM plants occurs at reduced level. Okay, then examples for C3 plants are rice, wheat, C4 plants, sugarcane and corn, CM plants, cactus, and pineapple. Okay, so we already finished with all pathway involved in the dark reaction or light independent reaction. So photorespirations again is a process in the metabolism of plants in which RUBP combines with oxygen with the help of enzyme Rubisco instead of carbon dioxide during normal photosynthesis. Because of the photorespiration, so the plants adapted to into a few pathways. So we have a carbon cycle for the all plants, but for C4 and CM plants, the adaptation is a hash slack pathway. Okay, so you should know the similarities between the pathway. Okay, I give you similarities of C3 and C4 plants. C3 and C4 plants both involve in the carbon cycle, both involve RUBP as carbon dioxide acceptor, both involve RUBP carboxylase to catalyze the fixation of carbon dioxide. But the difference is the enzyme for the carbon dioxide fixation is a PEP carboxylase for the C4 plants and CEM plants. Okay, what else? Okay, so you need to understand that in the, okay, alternative mechanism of carbon fixation occurs in C4 and CAM. Direction in C4 plants occurs both in mesophyll and bundle shift cells. Carbon dioxide fixation catalyzed by PEP carboxylase and until they enter the carbon cycle. Okay, while CAM mechanism operates in plants living in hot climatic conditions, and these plants both C3 and C4 cycles occurs in the mesophyll cell. So during the night, stomata open and allow carbon dioxide to diffuse into the mesophyll cells. CO2 will join PEP to form oxaloacetate. Then reaction catalyzed by PEP carboxylase and CM plants. Okay, for CM plants here, after that, oxaloacetate is reduced to malate, require NADPH from the light reaction. The accumulation of malate will increase the acidity in cytoplasm and this will influence the cell activity. Hence, malate is stored in vacuoles in the nighttime in the CAM plants. During the night, stomata open and starch is converted into PEP. PEP will combine with CO2. So okay, so that's all for. Sorry. Okay, so malate is stored in the vacuoles in the night time. So in the daytime, stomata of CM plants closed. No CO2 enters the mesophyll cells. Malate in vacuole will split into CO2 and pyruvate. CO2 is released for the carbon cycle. CO2 will join RUBP in the carbon cycle in order to produce organic compounds such as glucose. Then malate decarboxylation will also produce pyruvate. Okay, when malate decarboxylate carbon dioxide, CO2, they will also produce pyruvate. Pyruvate is converted into PEP by using 1 ATP, and this process known as phosphorylation because they consume a phosphate group from the ATP. And then the PAP produced is immediately converted into starch and stored. The reason PEP is converted into starch is to prevent PEP from competing with RUBP for CO2. So as I told you earlier, during the night, stomata open and starch converted into PEP. PEP will combine with CO2. Okay, so normally photosynthetic cells in C4 plants have large vacuoles. One of the major functions of large vacuole is store acid. All right. So for the during the day when the stomata are closed, malate undergoes decarboxylation to release CO2. This is to keep the internal concentration of CO2 high enough to prevent photorespiration. So remember comparison between C4 and CM plants. If you can find the difference, uh, cell involved in C4 plants are mesophyll cells and bundle shift cells, whilst the AM only involve mesophyll cells. Okay, type of separation in C4 plants, separation space means spatial separation space means decarboxylation occurs in bundle shift cells, but CO2 fixation occurs in mesophyll cells. Okay, while in the CAM plants, temporal separation means a time phase separation. So decarboxylation occurs during the day, while at the night, CO2 fixation occurs. Photorespiration in the C4 plants are low, while CAM plants has a moderate photorespiration. So the similarities between C4 and CAM plants, both pathway use PEP carboxylase to fix CO2, and both pathways use RUBP carboxylase in the Calvin cycle. RUBP carboxylase is a Rubisco. Okay, that's all for the dark reaction. I already covered of carbon dioxidization in the C3, C4, and CAM plants, and examples of the plants involved in this pathway. So what you need to do is do revision about this topic. Okay, so please do the worksheet, and we will see again in the next class. Okay, I think I will stop now. That's all for today. Bye.